The overall goal of this erythrocyte depletion method is to achieve a quick and simple leukocyte enrichment from a small blood sample.
Main advantages of this technique are that it is cheap, reliable and fast. Moreover, it can be applied to human and non-human blood samples.
Before beginning the lysis procedure, adjust the pipette for water administration to 2 milliliters and the pipette for PBS to 5 milliliters. Then place open flasks of water and PBS under a laminar flow hood at room temperature.
As the hypertonic lysis is a time-critical process, everything should be prepared previously. Moreover, the lysis procedure should always be done in the same way to have comparable results.
Next, transfer 100 microliters from each blood sample into a 15 milliliter conical tube and add 2 milliliters of distilled water to each sample with vortexing at the medium setting. Rest the cells for 60 seconds. Then add 5 milliliters of PBS and mix the samples by vortexing. Now collect the cells by centrifugation and decant the supernatants. Invert the tubes and tap the openings on a paper towel to remove as much liquid as possible. Then repeat the hypotonic lysis procedure as just demonstrated. After obtaining a dry pellet,